and we will continue our discussions the clinical chemistry. So our topic today is the urea cycle, and please remember that in the descriptions of each video of a clinical chemistry it will be a link of the PDF of this PDF. You can buy it just in $1. So let's start with the urea cycle. So the urea cycle, it's an important to know that the urea cycle, it's an important to get rid of the ammonia, which is the results of the protein degradations or protein metabolism. So we have here ammonia, we can say ammonium, sometimes NH3 is a plus CO2 plus ATP, or we can say NH4 plus HCO3 plus also ATP. So this is ammonia and NH3 plus CO2 plus ATP, and by the enzyme is called carbamyl phosphate synthase enzyme. This is the enzyme of it, is called carbamyl phosphate synthetase enzyme 1, because we have two types, and that is lead to the formation of the carbamyl phosphate. Okay, this reaction that happened only in the mitochondria of the hepatocytes of the liver cell. So this reaction, I see here, I said mitochondria because it happens in the mitochondria of the hepatocytes. The other reaction that is happening in the systole of the hepatocyte of the liver, I mean here. So we have ammonia and H3 plus CO2 plus ATP with the help of the enzyme called carbamyl phosphate synthase enzyme 1 that lead to the carbamyl phosphate. The other thing that I have to discuss is that the carbamyl phosphate synthase enzyme actually is the rate limiting enzyme, because I said before that we have to know what is the rate limiting enzyme to understand it. So we have ammonia, CO2, and by the help of the ATP, by the help of the enzyme carbamyl phosphate synthase enzyme 1, and that is lead to the carbamyl phosphate. This is the first information. The other information that there is a important cofactor that activate the carbamyl phosphate synthase enzyme, which is called N-acetyl glutamate. So the N-acetyl glutamate is an important cofactor that activate the carbamyl phosphate synthase enzyme to formation of carbamyl phosphate. And according to the N-acetyl glutamate, it's form of a glutamate plus acetyl coenzyme A, and that is lead to the N-acetyl glutamate plus CO. So what is important, that the N-acetyl glutamate is a cofactor of the carbamyl phosphate synthase enzyme, which is the rate limiting enzyme of the urea cycle. So carbamyl phosphate, and then we enter the systole of the hepatocyte, the arginine, and by the arginine transcarbamylase enzyme that is lead to the formation of the citrulline. So again, we have carbamyl phosphate go outside the mitochondria of the hepatocyte, and with the arginine, and we use the arginine transcarbamylase enzyme that is lead to the formation of the citrulline, and the citrulline with the aspartate, and by the enzyme is called arginocyssinate synthase enzyme, and that is lead to the formation of the arginocyssinate. And then by the arginocyssinase, that means there is a lysis that is lead to the production of fumarate and the arginine. And then the arginine by the arginase, that is lead to the formation of the arginine, or the arginine can lead to the urea. And then normally the urea should be excreted in the kidney and in the urine. What's so important about the urea that is composed of the carbon, which is make the double bond of the oxygen, and then binds to the two amino group NH2 and NH2. So it's a waste product. And why we say the urea is the waste product? Because the carbon is bind to all this stuff, binds to two ammonia and two oxygen, so no need to use the carbon, no need to use it. It's absolutely a wasted product, so it should be excreted in the kidney. So again, we want to discuss again what is the urea cycle. So we use ammonia, CO2, by the help of the enzyme carbamyl phosphosynthase enzyme 1, and that is a rate limiting enzyme of the urea cycle. By the cofactor is called N-acetyl glutamate, and then that is lead to the carbamyl phosphate. And then we enter this stall of the hepatocyte. The carbamyl phosphate with the orthionine, by the orthionine carbamyl trans, or we can say transcarbamylase, and that is lead to the formation of the citrulline. And the citrulline by the aspartate formation arginocyanate synthase, because what is related of the arginocyanate with the citrulline and the aspartate? Because the product is called arginocyanate. So citrulline, aspartate, arginocyanate synthetase enzyme that is lead to the formation of the arginocyanate, and by its lysis that is lead to the formation of the arginine and the fumarate. And then the arginine that is lead to the formation of the urea, which is the waste product and should be excreted in the kidney. We want to know actually what is happened here in the muscles. The muscle, we have alpha ketoglutarate, and by the alanine transferase enzyme that is lead to the formation of a pyruvate. And the same enzyme alanine by the alanine transferase enzyme that is lead to the formation of a glutamate. And then the glutamate, it will be go through the blood vessels and enter the liver. The glutamate that is lead to the formation of alpha ketoglutarate, and by the glutamate dehydrogenase enzyme and the help of H2, that is lead to the release of the NH3, and then the NH3 by addition of NH4 that is lead to the NH4, and then the urea cycle continua. Okay, so now we know what is the source. It comes from the muscles because it leads because of a protein degradations. So it starts with the glutamate, and then the glutamate turn to the alpha ketoglutarate. But I want to talk about this reaction because it's reversible reaction. The glutamate can turn to the alpha ketoglutarate, and by this reaction that is lead to the production of the ammonia, which is a toxic thing. But if it's reversibly, because it's reversible reaction, it should be arrow here, glutamate dehydrogenase acts reversibly. So the alpha ketoglutarate trap NH3 or ammonia or whatever by the help of the glutamate dehydrogenase, and it will turn it to the glutamate, and the glutamate itself, it's also trap another ammonium group with the ATP, and that is turned to the glutamine, which is acid, and excreted in the kidney. This reaction, it will be very, very helpful to get rid of the ammonia group in our bodies. But if it's started with the alpha ketoglutarate, so the alpha ketoglutarate again trapped NH3, use the NH3, and by the help of the glutamate dehydrogenase enzyme, and that is turned to the glutamate, and then the glutamate trap another NH3 and turn to the glutamine, and then it excreted in the kidney and the urine, and we get rid of this NH3. But if it's in this direction, then that is lead to the formations of an NH3, and we use here the same enzyme. Okay, now I'll talk about if this NH3 and H4, there is a defect here in the liver, then the NH4 go to the bloodstream, And if it's go to the bloodstream, then it's form more of the water, more of the water, more of the water here. And if it's go to the brain, I said here is a brain, then the NH4 and the glutamate, and by the glutamate or glutamine synthesis enzyme that is turned to the glutamine. And you know what is a problem of the glutamine? It increases the osmotic pressure, and then the glutamine turn or suck all the water that is split in the bloodstream and becomes only a brain, and that is lead to the cerebral edema, and that is increase the intracranial pressure, and that is cause hernia and comatose. So that is the problem here of increasing the glutamine here, because now we understand how the glutamine lead to the formations of the ammonia, because you know the ammonia which is so important, that is increase the level of glutamine and the glycine. And what is the problem about the glutamine? That increase the osmotic pressure and just to trap and sucks all the water of the body. So that is a problem. Again, an H4 lead to the formation of more glutamine and more glycine. Okay, so we here again we have a protein, and by the degradation of the protein, that is lead to the ammonia to turn to the urea. All that is happens on the liver. The first reaction till carbamyl phosphate, that is happen in the mitochondria of the hepatocytes, and the rest it will be in the systole of the liver. Then it will form the urea, and it should be excreted through the kidney. We have to know that the rate limiting enzyme is carbamyl phosphate synthetase 1. Again, the rate limiting enzyme is called carbamyl phosphate synthetase 1. The cofactor is N-acetyl glutamate. Okay, we discuss also after the carbamyl phosphate and with the orthionine, and then the enzyme is called orthionine transcarbamylase enzyme, and that is formation of the citrulline. Here this reaction, the orthionine transcarbamylase enzyme, some deficiency that is can maybe happen, and that is lead to the formation of the orthionine transcarbamylase enzyme deficiency disease. And what do you think? So the citrulline is reduced. Okay, and if it's reduced, then the orthionine reduced also, and that is lead to the increasing of uric acid, and carbamylase phosphate also increase, and that is lead also to the production of the ammonia. And the orthionine transcarbamylase deficiency is X-linked disease. The symptoms of hyperammonia, that is lead to the asterixis and cerebral edema, and we discuss how the cerebral edema occurs. Now we talk about the treatment of this. The first one is decrease the protein intake. That's mean we decrease the cause of the urea cycle, with the cause of the urea formation, and by decreasing the protein intake, and also decrease the ammonia formation by two things: lactulose, and by increasing the GID excretion. The lactulose increase the excretion of ammonia, that's trap the ammonia and get rid of it through the stool. And we have the rifaximin, and that is lead to decrease the colonic production, destroy the bacteria. But the question is, what is the bacteria? Why we mention here the bacteria? What this does? So we have the urea. Actually, there is in a human urease a production bacteria or producing bacteria. There is a bacteria that is present in our colon, in our GIT, that produce the enzyme called urease. And the urease, that is lead to the degradations of the urea into ammonia and CO2. So the bacteria lead to the formation of the ammonia. The human itself, there is no urease, but the bacteria inside him, like a Helicobacter pylori, which is, we studied this before in the stomach, produces urease enzyme, and the urease turn the urea into ammonia and CO2. So if we do something like called rifaximin, that is lead to destroy this bacteria that is produce the urease enzyme, which is turn the urea into ammonia, so we avoid the ammonia production. So we have two solutions, the lactulose and rifaximin. The lactulose increase the excretions of the ammonia through the GIT. Rifaximin destroy the bacteria that is produces the urease, which is turn the urea into ammonia. So that is all about the urea cycle. Thank you so much for listening.